Welcome to our presentation on the kidney nephron. Our objectives are A, give a large label diagram of the kidney nephron. B, with respect to microscopic structure, describe the functions of the nephron in relation to ultrafiltration, absorption, and secretion.
So as you can see, here's a large diagram of the kidney nephron. And blood is being taken through the afferent arteriole and is carried through the glomerulus. So the glomerulus is basically a cluster of blood vessels that shoot small substances into the Bowman's caps. This is then pushed to the PCT, or the small convoluted tubule, and it goes down to the loop of Henle where sodium and water is reabsorbed. Sodium and water is being reabsorbed also in the DCT, which is the distal convoluted tubule, and this is then passed to the collecting duct. The urine then flows to the renal pelvis and into the ureter.
The nephrons are the subunit of the kidney that deals with all the filtering. In filtration, about 120 milliliter per minute get pushed through from the blood to the capsule, and the 1% of which remains removed from the body, and the rest is reabsorbed back into the body. So of all the things that gets pushed out, we want to know what get pushed out and why. So this is a result of 3 filtration membrane. First, you have the filtration membrane that comprises the blood vessel itself, which is the endothelial. The endothelial at the glomerulus has a hole in it, which is referred to as the fenestrated endothelial. So the fenestrated endothelial, which is the first filtrate membrane, has a width of 70 to 100 nanometer, which determine which substance are able to pass through. For example, red blood cells, which are 10 micrometer large, will not be able to pass through. So what are going to pass through the fenestrated endothelial are things such as protein, ions, glucose, and fluids, etc., which are small. So the next filtrate membrane will be the connective tissue called the basement membrane, which is filled with collagen. And one thing that you should know about collagen is that it is negatively charged. Know that the protein which will be flowing through is also negatively charged. Therefore, the like charges will repel each other. So with the basement membrane be negatively charged, because of collagen, it will repel protein molecule. So the protein will not be able to pass through the membrane. Now you have the third filter membrane, which is known as the podocyte, which lets some substance and particle pass through, which is about 10 to 40 nanometer, which is very small. With this small size, it will stop all cells and small protein molecules that may have gotten through the basement membrane. Therefore, protein, cell, etc. will not get filtered out of a healthy human body.
So the function of the nephron in relation to absorption. So reabsorption is the movement of water and solutes from the tubule back into the plasma or into the circulating blood. The bulk of reabsorption takes place within the proximal convoluted tubule, or the PCT, without being under hormonal control. And more than 70% of the filtrate is absorbed here.
Table showing substances that are reabsorbed in the nephron and their locations. In the proximal convoluted tubule, almost 100% of glucose is reabsorbed by the secondary active transport with sodium ion. Oligopeptides, proteins, amino acids, almost 100% is reabsorbed, symport with sodium ion. iron. Vitamins are reabsorbed, lactate are reabsorbed, urea 50% is reabsorbed by diffusion. Sodium 65% is actively reabsorbed. Chloride is reabsorbed, symport with sodium ion and diffused. Water 67% is reabsorbed osmotically with solutes. Bicarbonate is reabsorbed and diffused, and 80% to 90% symport reabsorbed with sodium ion. And potassium 65% is reabsorbed and diffused. Calcium is reabsorbed and diffused. Magnesium is reabsorbed and diffused. Phosphate 85% is reabsorbed, inhibited by pyrethroid hormone, and is diffused.
For the loop of Henle, we have sodium 25% reabsorbed in thick ascending limb and active transport. For chloride, it's reabsorbed in the thin and thick ascending limb, diffusion in the ascending limb as well. For water, we have 50% reabsorbed in the descending limb and osmosis taking place. Bicarbonate, we have reabsorbed symports with sodium and antiports with chloride in ascending limb. We have HCO, reabsorbed diffusion in ascending limb. We have potassium, 20% reabsorbed in thick ascending limb and symport. For calcium, reabsorbed thick ascending limb and also diffusion. Magnesium, reabsorbed in thick ascending limb.
In the distal convoluted tubule, 5% of sodium is reabsorbed by active transport. Chloride is reabsorbed and diffused. Water, 8% is reabsorbed if ADH and by osmosis. Magnesium is reabsorbed and phosphate is reabsorbed and diffused.
For the collecting ducts, we have urea reabsorption in medullary collecting ducts. 5% is reabsorbed and stimulated by aldosterone active, that's for sodium. For chloride, it's reabsorbed and symport. For water, we have variable amounts reabsorbed and controlled by ADH. We also have bicarbonate reabsorbed antiport with chloride. We have HCO reabsorbed antiport with sodium. And we also have calcium reabsorbed if parathyroid hormone is present.
The function of the nephron in relation to secretion. The transport of materials from the peritubular capillaries to the renal tubular lumen is known as renal tubular secretion. The movement of water and solutes during secretion is governed by the process of osmosis, diffusion, and active transport. Active transport and diffusion are the primary causes of secretion. Only a few chemicals such as hydrogen ions, creatine, urea, excess ions, and drugs are usually released. And these are usually waste products. Tubular secretion happens throughout the nephron, beginning at the proximal convoluted tubule and the distal convoluted tubule. Overall, tubular secretion is the removal of substances from the blood into the renal tubules, leading to the production of urine. So you have two secretion takes place in two sections of the nephron, that is the proximal convoluted tubule and the distal convoluted tubule. So starting with the proximal convoluted tubule. So the filtrate from the Bowman's capsule is introduced into the proximal convoluted tubule. The secretion process begins at the proximal convoluted tubule, as said before. So the proximal convoluted tubule is comprised of simple cuboidal epithelial cells with a pronounced brush border of microvilli. The microvilli have a wide surface area, which allows for maximum solute secretion of the filtrates because these cells promote active transport of solutes across membranes. A great number of mitochondria are required to make enough ATP. Secretion of hydrogen ions occurs through secondary active transport through the apical surface / membrane.
Secretion occurs in the distal convoluted tubule at the last segment, where most of the filtered solutes and water have returned to the blood vessels by this time. The distal convoluted tubules include two types of cells: simple cuboidal epithelial cells, which is composed of the intercalated cells, and principal cells. The principal cells secrete potassium ions, while the intercalated cells secrete hydrogen ions, consequently performing its function in blood pH regulation. The potassium ion leakage channels facilitate the exit of potassium ion into the tubular fluid. There are fewer microvilli, and these cells are less active than those found in the proximal convoluted tubule. These cells, however, have a significant number of mitochondria because they must pump ions against a concentration gradient.
Thank you for tuning into our presentation. And hello and welcome back to Battle of the Nurses. We have with us today Team Cortex, that is Akila Stanley and Shamila Richards, and Team Medulla, that is Rashana Morris and Janelle Graham. So first question, what is the function of the microvilli?
The function of the microvilli is to provide large surface area to facilitate reabsorption.
That's correct. Team Medulla, what does the tubular where does tubular secretion begin?
In the distal part of the nephron?
That's incorrect. It is the proximal convoluted tubule. Team Cortex, what substances are reabsorbed in the distal convoluted tubule?
Calcium, sodium, and chloride.
That's incorrect. Oh my God, the 2 minutes run out. Continue.
Mhm.
2 minutes run out fast. Hello, welcome back to Battle of the Nurses. We have with us today Team Cortex, that is Akila Stanley and Shamila Richards, and Team Medulla, that is Janelle Graham and Rashana Morris. Sorry. So, Team Cortex, what substances are reabsorbed in the distal convoluted tubule? List three of them.
Calcium, sodium, and chloride.
That's incorrect. Sodium, chlorine, and water. So Team Medulla, what are the two cells that make up the distal convoluted tubule?
Intercalated cells and the principal cells.
That's correct. Team Cortex, what is the function of the microvilli?
The function of the microvilli is to provide large surface area to facilitate reabsorption.
That's correct. Team Medulla, what are the three filtration membranes?
The three filtration membrane are the fenestrated endothelial, the basement membrane, and also the podocytes.
Okay. Team Cortex, what are the two parts of the loop of Henle?
We have the ascending and descending limbs.
Finally, Team Medulla, what does the tubular secretion, where does tubular secretion begin?
In the distal part of the nephron.
That is incorrect. It is the proximal convoluted tubule. So the first in history of the Battle of the Nurses, we have a tie between Team Cortex and Team Medulla. So we have a bonus question. So what substances are reabsorbed in the proximal convoluted tubule?
We have water, we have sodium,
Sodium,
we have glucose, amino acids,
That is correct. And the trophy, where's the trophy? The trophy goes to Team Cortex.